This past year has been absolutely an incredible journey for ASM. And as we've set the stage for ASM's new strategic roadmap, it has been inspiring to watch the pieces begin to fall into place as we reimagine the way that ASM will engage with the world. A key theme I've taken from ASM's new strategy is that it is woven into every element of our new vision is the idea of science in service of humanity. We talk about harnessing the power of microbes to solve humanity's biggest challenges, but what exactly does that mean? This past year, serving as your ASM president has been an honor to watch all of the amazing work that you do, from confronting the challenges of climate change to devising strategies to combat antimicrobial resistance, from exploring potential of microbiome to driving innovations in the bioeconomy. You have all played an integral role in paving the way towards a healthier and more sustainable world. These examples are just a glimpse of the impactful contributions microbiologists are making to shape a brighter future for us all. In this forum, we're going to dive into the concept of using microbial sciences to change the world. We'll hear about the innovative research that our three presenters are working on and see how their work could directly be tied to serving humanity. Whether it's conducting fundamental mechanistic discovery research, collaborating with biomedical engineers, climate scientists, ecologists, and other partners to develop workable solutions using microbiology, or serving as advocates for those sustainable solutions, microbial sciences have many roles to fulfill, leveraging our scientific knowledge to influence and shape our world. To address the biggest challenges of our world, though, we need a fundamental understanding of microbial communities, their impact on the environment, and how these communities influence human and animal health, our soil, our water, our air, as well as food production. What happens if these communities are disrupted? Or if these members of these communities go extinct? Knowing that microbes are nature's best chemists, how can we tap this knowledge to build a circular bioeconomy, combat pollution, or combat climate change? We need to understand how changes in the distribution of pathogen vectors and reservoirs impacts disease spread and help anticipate where the next pandemic will come from. How can we use this knowledge to develop detection and countermeasures? There are many more fundamental questions like this, questions that we as microbial scientists are constantly thinking about. As the conditions of our world change, we're continuing to re-examine, build upon, and interpret our previous understanding of the data that we gather. After all, science is an iterative process. Our three speakers tonight will be highlighting aspects of fundamental discovery microbiology research and make connections to how this knowledge is or could be used to develop solutions to global challenges we face. And now I'm honored to introduce to you the speakers that we have today. First will be Timothy Donohue. He is the director of the Great Lakes Bioenergy Research Center at the University of Wisconsin-Madison. He is a fellow of the American Academy of Microbiology, as well as the American Association of the Advancement of Science. And he served as president of ASM in 2013. His laboratory's research analyzes pathways and networks that microbes use to grow, generate biomass, or produce fuels for sunlight or other renewable sources of energy. Our next speaker, Emiley Eloe-Fadrosh, is an award-winning researcher and lab lead at the National Microbiome Data Collaborative Joint Genome Institute. She received the 2020 Berkeley Lab Directors Award for Exceptional Early Scientific Career Achievement. Her current research focuses on leveraging thousands of metagenomic databases from human-associated and environmental samples to identify novel microbial life as well as viral diversity. And our last speaker will be Nat Moorman, who studies human cytomegaloviruses to define new targets for the development of novel antiviral drugs, and he's in the Department of Microbiology and Immunology at UNC Chapel Hill. Moorman's lab uses a combination of new technologies and traditional molecular virology techniques to investigate host pathogen interactions between HCMV and the infected cell. So thank you for your attention, and now it's my great pleasure to turn the podium over to our first speaker, Tim Donohue.
I want to thank Virginia and the Society for giving me this honor. It's really great for me to talk to you today about what I see as a vision for how microbes can act as catalysts for a new bio-revolution. So I'm going to divide my talk into two parts. Very quickly, I'm going to remind everybody how microbes have revolutionized our world. This is kind of like me bringing coals to Newcastle for this audience. And then I'm going to spend most of my time talking about how I think microbes can contribute to a bio-revolution after I define what that is. So as we heard on Thursday night with READDI, this was a great kickoff that microbes have really created the world that we live in. If you think about what goes on in the oceans, it's a major sink for CO2. They provide all the oxygen that we need to breathe. And all of this is powered by renewable resources. Without those activities, we wouldn't be here today. And hopefully you'll hear from Emiley later how that's an important part of what we need to preserve as we move to the future. This is another slide that I've thrown together some old vignettes to remind us that there is a long history of microbial catalysts benefiting us today. Whether it's on the left, over 100 years ago, the discovery of root nodule bacteria being able to be used as bio-based fertilizer instead of using ammonia. Something close to my current interest of biofuels, as we think of those as new things, but really Henry Ford over 100 years ago predicted that all of his cars coming off the assembly line would be running on bio-based ethanol. That didn't happen, but we're still in that business today. Then another two of my favorite pictures on the right. One shows Mary Hunt, a scientist at the USDA labs in Peoria, out at the farmer's market in Peoria on a Saturday morning, picking up a moldy cantaloupe, and that turned out to be the Streptomyces strain that was heavily mutagenized, but created all the penicillin that was used to treat wounded soldiers in World War II. And then at about the same time, this University of Wisconsin gal, according to the New York Times, Elizabeth McCoy, having a patent to improve the production of bio-based butanol. It wasn't necessarily the strain, but it was the combination of the strain and Dr. McCoy at the time figuring out how to make that strain phage resistant so that it could grow in industry and not have the fermentations get wiped out. So there's phage biology, there's root nodules, there's everything that we all think about today. Today, we know a lot about microbes that are involved in making food and chemicals, both pure cultures on the top, and actually there are synthetic communities of microbes, synthetic microbiomes that are involved in making soy sauce. And then moving over, we've already heard a lot this week about genome-enabled antimicrobials, and something close to my heart I'll call recombinant drugs. As a first-year graduate student, I will never forget hearing a talk by Paul Boyer talking about recombinant DNA technology and the potential it had to produce insulin in bacteria sometime in the middle of the 21st century. But instead, that was available in 1980. And a lot of us are here today because of a molecular recombinant RNA vaccine that we've possibly taken in the last couple of years. So with that as a backdrop, what is this biorevolution? And how can those microbial activities I just talked about help catalyze that? So this is not a new idea. It goes all the way back to the Bush administration in his 2010 State of the Union address. He talked about how biology could revolutionize industry. It's been accepted by the UN in its 2030 Sustainable Development Goals, and it's been reinvigorated by the Biden administration in its biotechnology and biomanufacturing reports. But in the middle of that, and in the pictures, are one of my favorite reports from the McKinsey Group. Leave the politicians aside and look at their vision for up to 60% of the inputs to the global economy could in principle be produced biologically. Everything from agriculture, energy, materials, health, and consumer goods. So that's what I want to spend the rest of my time talking about today. So let me move on now to talk about a vision for how microbes can contribute to a bio-revolution. And I like to use this slide to say that we have the ability here. You're in the best society in the world to be thinking about this because microbes can really be in the middle to catalyze this. On the left-hand part of this cartoon shows a large number of diverse renewable raw materials that can be used to power this biorevolution. Everything from non-food agricultural materials to industrial residues, municipal waste, CO2 and syngas can be converted into a suite of products on the right of new or replacement or even improved chemicals, fuels, and materials for society. And in the middle of this are your friends, my friends, microbes. Because they, after all, as Virginia alluded to, have been doing chemistry longer than people. And they have the potential to be the catalyst in these biorefineries, either pure cultures or communities. And I'll give you some examples as we move on. The first example is the use of what we call, or I call, energy crops to go into a biorefinery to make fuels and chemicals. And that perspective comes from a group that I have the pleasure to lead called Great Lakes Bioenergy. Great Lakes Bioenergy is one of 4 bioenergy research centers funded by the Office of Biological and Environmental Research at the Department of Energy. The other 3 partner institutions are up on the top of this slide. We all have a common goal to remove the barriers to microbial conversion of non-edible or so-called lignocellulosic biomass into hydrocarbon fuels, products, and materials. So let me now say a few words about what that really means. So what that really means is the country needs to be able to produce the lignocellulosic biomass that we're going to feed the microbes to turn into these products. So how feasible is that? Well, here's an atlas that we've developed in Great Lakes Bioenergy that shows land that's available to produce non-food biomass across all 48 states in the US. And it's not imaginary. It's informed by historical paper, productivity records, as well as satellites. And if you want to go to that website that's up there, you can zoom into any part of the country that you live in and look at productivity at 30 by 30 meter scales, as well as looking at it nationwide. There are sites on this map that are yellow or green that have few limitations on their use, and others that are orange and red that have more because they're mountains or they're national parks or they're conservation areas. So this is a roadmap for decision makers to figure out where that land is available. And as I said, you can go in and overlay what the yield data, whether it needs to be watered, and whether it's dedicated for conservation. So with that as a backdrop, what do we want to do with it? So we're developing so-called bioenergy crops that are dedicated non-food plants, things like switchgrass, poplar, or sorghum. You may have heard about those. And the DOE and the USDA estimate, based on the kind of data I showed on the previous slide and other data, that there's over 1 billion tons of that available in the US every year. And the responsible use of a fraction of that could reduce US fossil fuel use by 30%. So that's why on the right-hand side of this slide, I've showed one of the barrels as a green barrel that's powered by lignocellulosic biomass. And that includes products that are hydrocarbons for so-called sustainable aviation or jet fuel, shipping and marine diesel, and even cars for as long as we're going to be driving them. And that's the red, orange, and blue part of the material in the barrel, plus chemicals, which is the yellow slice on the top. And if done right, with the right crops on the right land and the right microbes in the right soil, we have modeled that this can reduce the net greenhouse gas footprint of this activity by 3 to 5-fold if you go from field to product. So now let me tell you a little bit about the microbes that are going to be the catalyst in this type of a revolution. So I'm going to walk you through 2 types of microbes that we're using in the beginning part of this. If I gave you a ton of a bioenergy crop, you would create it into 2 types of juices or liquids. One on the top is a sugar, polysaccharide, or cellulose fraction, and the other I'll come back to in a minute is a lignin fraction that's the source of aromatics. So in this first slide, we've developed and continue to develop and use genome-enabled data to engineer industry-ready yeast and bacteria to take those polysaccharides on the top, which is 60% of the biomass, and convert it into approved fuels or precursors for upgrading. And our favorite fuel molecule in Great Lakes Bioenergy up there on the upper right is isobutanol, an alcohol that's already been approved by the EPA for blending and with diesel and aviation fuel, and which can be polymerized to actually replace a large fraction of the fossil fuels that goes into those engines. We're very proud of what we've accomplished with this so far, but we're not there yet. We still need, on the lower right, to go into the techno-economic analysis of this so the production of isobutanol is cost competitive with field-to-fuel processes. Because you don't want to fly home on a plane that costs you 10 times more because you're using Tim's isobutanol. It needs to be cost competitive. And we need to improve that then to increase the rate, tighter in yield of this pipeline with different bioenergy crops, because switchgrass is not the same as sorghum and it's not the same as poplar, and the microbes need to be able to handle whatever you feed them. And making isobutanol is not something that these microbes have evolved over billions of years to do, so there's off-target effects that we need to account for as we move on. So how are we going to improve that? One of the big problems is sugar utilization of the yeast and bacteria that we're using. So here's a paper that was just published in a non-ASM journal where they looked at sequences of over 1,100 yeast strains and grew them under 24 different conditions and did genomics on it to understand the metabolic diversity of this large panel of potentially industrial yeast strains. And one thing that's come out of that right away is that they can use machine learning to identify alternative pathways for sugar utilization. In this case, it's galactose, but I'll come back to this point in a moment. So here's an example of things we can do today that we never would have been able to do in the pre-genomic area with machine learning and with good old-fashioned biochemistry and genetics. Let me now move on and talk about the other polymer that I mentioned. That's lignin, which is a heteropolymer of aromatics. It's about 30% of a ton of biomass that I might give you. And lignin is shown on the top of this slide, and it's this, for lack of a better word, abysmal-looking polymer that's composed of red, green, and blue subunits that differ only in the amount of methoxy groups on the ring. And when we first started, people said, you can make anything you want out of lignin except money. So don't try to do anything with it. Well, we've now developed ways to break it down into its component parts. And depending on how we break it down, you get a ring, but you get these different substituents hanging off the top of it at 12 o'clock. So what are you going to do with all of these mixed aromatic compounds? What you need is a champion microbe that can take all of those and funnel them through a metabolic pathway to make valuable compounds. And we found one, or else I wouldn't be telling you this story. And that's this organism called Novus Fingovium aromaticivorans, which was actually isolated from a polyaromatic hydrocarbon contaminated site in the Savannah River Basin in South Carolina by previous DOE researchers. And that's it on a Petri plate. And it has in its genome the ability to eat a lot of aromatic things, because that's what it was doing in Savannah before somebody scooped it out of the river. So what have we done with that? We've been able to take this organism, and by a combination of enzymology, genetics, and genomics, we've been able to interrupt its metabolic pathways to produce that molecule on the upper right, pyrrone dicarboxylic acid, which is a bio-based precursor for biodegradable polyesters, adhesives, and antimicrobials. So that's cool. What else could we do? By a different set of genetic mutations, we wanted to make this other linear dicarboxylic acid, cis-muconic acid. And if you look at PDC, and you cleave that ring, it's pretty easy to think how it's going to open up. But it was a lot of work to get there. In the beginning, we thought we were going to have to import new enzymes into this organism to do it. But as we began to build it, we realized that the organism already had its own enzymes. They just weren't expressed. So we purified them, confirmed they were active, and put them in another spot in the genome where they were expressed. And now we can make CCMA at over 100% yield in biomass from biomass aromatics. And you're probably saying, what is this guy telling me? You're getting more product than the food that you're giving it. And that's because in the left-hand part of this graph, we don't understand everything that's in the biomass food that we feed them. But the things we can track are all going away, and we're getting over 100% product yield. So that's cool. So what else can we do with this organism? So it's yellow, and to a microbiologist, yellow means it's making a pigment. And the genome predicted that pigment was a carotenoid or an isoprenoid. So we have also gone in now and rewired its carotenoid pathway to make 3 different strains that are actually different colors on those plates. One of them is in your eye drops if you have dry eyes. One of them that looks orange is beta-carotene, which you know, that's from carrots. And one of them, the pink one, is a compound that aquaculture feeds to salmon. So when they raise farm salmon, you get pink salmon. Those 3 Petri plates, the molecules in there represent over 60% of a multi-billion-dollar a year carotenoid industry. So we can also make carotenoids from these organisms. But if you code, we can put an AND gate into this strain, because we can make strains that secrete either one of those products and any one of those colored compounds. So now from one reactor, you can get 2 products. So we call these multitasking microbes. They're able to do 2 things at a time, which for me sometimes is hard. So the last couple of examples I want to talk about move away from pure cultures and move to communities to convert fermentation residues to chemicals. So you might say, what are you talking about, fermentation residues? So on the top of this slide, I've compared a couple of fermentation broths that you may have experienced at one point in your life to a bottle of lab alcohol, and it looks very different. Well, when you do any of the fermentations that we do, the same thing happens. You get an alcohol product, but there's leftover carbon in the residue. And a lot of that carbon happens to be sugar. So what are we going to do with that carbon? Well, you could throw it away or burn it, but Mother Nature has put a lot of energy into making that carbon, so we want to do something better with it. But the fermentation microbes that we use can't eat it. So what do? We go to the wastewater treatment plant in Madison, Wisconsin, and we feed that fermentation residue to a sample of the wastewater treatment microbes that we domesticate in the lab. And out of it comes medium chain fatty acids, C4 and C6 fatty acids, whose revenue can actually help reduce the price of the fuel that you made in the first fermentation. And it also lowers the disposal cost and the greenhouse gas footprint. We can also go in with the help of people like GAGI to do metagenomic and metatranscript analysis of this that helps us predict pathways to move forward. So I hope I've convinced you that by getting at the sugars, the aromatics, and the residues, we can make products from all of the fractions. And that enables an industry that works just like the petrochemical industry, which makes money from chemicals and money from fuels. So we're not there yet, but let's assume for the moment that we can make one green barrel. How are we going to get to the other two? Well, it turns out there is an awful lot of organic residue spread out all over the country in this heat map. So what else can we use as a residue to make products? Well, I live in Wisconsin, and there's a lot of cows in Wisconsin. I'm going to briefly tell you a little story about fermenting your whey, W-H-E-Y, like cheese whey, into profits. So how feasible is that? Well, it turns out that in Wisconsin alone, we generate over 10 to the 7 metric tons of dairy residues every year, either as filtrates or wheys or even manure. And I'm going to tell you about using manure here in a moment. So what can we do with that? So one of the first things we took is we took this milk permeate, which is a filtered product that comes out of processing milk, and we fermented it the same way we did before, and we compared how much we made and what the theoretical yield was, and that green line that goes across the bottom is what a dairy would get for selling that on the market. And look at lactic acid. We're already doing better there for product, but we're not at theoretical yield yet. So how do we get there? So we got there by doing the same experiment with different mixtures of dairy residues, with solubilized manure, with going back to our fermentation residues. And out of that, we have well over 10 to the third high quality metagenome assembled genomes thanks to JGI. And that allows us to use machine learning to make predictions about residue by microbe, by product interactions. And by just following on those with one of those predictions, we now have fermentations with dairy residues that produce 75% of the total fermentation product is lactic acid. That's 40 grams per liter, and it does it continuously over 30 days. And we've kept them going even longer. So this shows the potential of genomics and microbes to power this revolution. So I'm going to stop and hopefully say that I've convinced you that microbial sciences on its own, collaborations with chemists, engineers, and computer people can create needed new products for society that will power a multi-trillion-dollar per year industry, simultaneously reducing net greenhouse gas emissions, providing jobs, STEM and other jobs for local communities that will generate resources that will stay where those raw materials are generated, and create economic justice for citizens, not only in the US, but if you're from overseas when you go home around the world. So with that, I'll thank you for your attention and pass it off to the next speaker.
Great. Well, thank you to Virginia and Stefano and the entire ASM team for this amazing opportunity. It's really an honor to be here tonight, and we've just had so many wonderful interactions with ASM over the years that I'm just so delighted to be here tonight. So I wanted to talk about ecosystems. Ecosystems from the environmental sciences perspective, ecosystems of data, and ecosystems of people, of the scientists, engineers, and software developers who study microbes, who use data to make inferences, and who take those inferences into action. But first, breathe in and breathe out. Join me again. Breathe in and breathe out. Thank a microbe, okay? Look at our incredible home, Earth. We have microbes to thank for the oxygen we breathe, specifically cyanobacteria that billions of years ago took the atmosphere from a reducing environment to an atmosphere that was oxygenated and that supports life as we know it today. And it's the microbes that are able to carry out this tremendous diversity of reactions that can transform metals, microbes that breathe metals, microbes that can fix nitrogen, microbes that can take organic matter and translate it back into carbon dioxide. And so, as you heard from Tim, microbes really are the drivers of Earth's biogeochemical cycles. But I feel like I'm preaching to the choir here. Because, of course, microbes, and as microbiologists, we've known this for some time. And so we know that microbes can be both the source and sink of the three main potent greenhouse gases: carbon dioxide, methane, and nitrous oxide. And so one of the challenges that we face as microbiologists is how do we take these small-scale microbial processes and scale them to larger models within the Earth system? Shown here is a report from the IPCC that shows the state of these three potent greenhouse gases increasing over the years. So as microbiologists, we really have an urgent calling to try to understand how these microbial processes can impact Earth system models and developing new models for how these microbial processes can be used, and also particularly partner with microbes for how we can understand the microbes that impact these processes. So I would offer that metagenomics expands our toolkit for how we can access microbial communities. We know that there is a large diversity, a large phylogenetic diversity of microbes can then not be pulled into cultivation and studied under laboratory conditions. And so how do we access these microbes? Across diverse environments, aquatic, terrestrial, we can use metagenomics to access the genomes within these environmental systems. And that these DNA can act as a proxy or a metagenomic blueprint for looking at these microbes. Of course, this has been done for over 2 decades. And so increases in the way and the throughput that we can use metagenomics to study microbes has really been improving. And as Tim mentioned, the use of metagenome assembled genomes, or MAGs, reconstructing population genomes and looking at the composite readout of those sequences can really help us to understand who is there and what they're doing. And that gives us also a view into both the community function and the metabolism of those microbes within the systems. And so we can use this data and the metabolic capacity that is encoded within the genomes to try to look at regional and global scale models. So when we look at diverse ecosystems, there are, of course, notable differences across environments, as you would expect. Shown here is a graph of sequence diversity on the y-axis as measured by Immerse, and the percent of reads mapped on the x-axis to reference databases and specifically NCBI's RefSeq database. So thanks to large investments by the NIH for the Human Microbiome Program, as well as the EU's MetaHit program, the gut microbiome is one of the best studied microbiomes right now. And so what I'm showing here is the data from a subset, obviously, of gut microbiomes showing that there's low sequence diversity and yet large reference databases that can be mapped to, and that has really advanced the field of human microbiome research. When we look at environmental systems, the story changes. So there's a much larger sequence diversity and less reference genomes with which to map to. And so shown here, again, apologies, not ASM journals, but for example, the Terra Oceans has been a great way, a great program to map marine microbial diversity in new ways and is improving the amount of reference data from aquatic systems. However, when we look at soil microbiomes and soil systems, it still remains this black box where you see very high sequence diversity, but a lack of reference data in the databases. And so that brings me to my research and my team at the Joint Genome Institute. So the Joint Genome Institute is an Office of Science, Department of Energy, user facility that provides genomic capabilities to the global research community. We use high throughput sequencing and expert analysis to provide information and data from microbes and viruses to microeukaryotes, all the way up to fungi and plants. A newer program that has been recently launched in the past couple of years is the National Microbiome Data Collaborative, an effort to democratize microbiome data to the research community. We use infrastructure, software, and data tools to provide researchers easier ways to create, use, and reuse data. So together, the JGI and the NMDC are joining forces to improve the availability of metagenomic data and multiomic data across diverse systems to improve this metagenomic blueprint with which the community can use to better understand microbial systems and processes. So the JGI has been in operation for 2 decades now, over 2 decades. And here's a picture of how sequencing technologies have changed over time. From the very beginnings of 454 and Celexa and the early days of Illumina to improvements over time with scale and throughput, from megabases worth of data to hundreds of terabases worth of data, which I think is very exciting. One of the other areas is looking at short read versus long read, shown here in the graph in blue and green. Long read applications for metagenomics is a really exciting way to begin to look at communities across diverse systems. But today, I'm going to focus on short read sequencing, which has really been the workforce for microbial ecologists over the past 2 decades. And so, as I mentioned at the beginning, I wanted to focus on ecosystems of scientists, of how researchers across and within organizational boundaries can work together and join forces to understand big, grand challenge problems like understanding soil microbiomes. And so we've been very fortunate to partner and team up with the National Ecological Observatory Network, or NEON, which is funded by the National Science Foundation. NEON is a continental scale, long-term user facility that focuses on using a distributed network model to collect standardized samples across their 81 field sites. What they do is do diverse sample collections spanning anywhere from airborne remote sensing down to soil samples for metagenomics. And this, I think, is a really exciting facility that in the biological sciences has never been done before. And so what we were specifically interested in was to look at the soil microbiomes. And they, over almost a decade, have been able to collect and sequence soil metagenomes and amassed a really large amount of soil metagenome data collected in standardized ways and provided to the global research community. What we were interested in doing was to leverage and go beyond that. And we started working with Kate Thibault and Hugh Cross at NEON to pilot how the JGI could generate even more data from these unique samples across the United States. So we worked with them, collecting 100 samples and combining this data to generate 5 terabases of metagenomic data from these diverse soil environments. For those of you who may not be familiar with the scale of data, 5 terabases, if we think about a single bacterial genome with a 5 megabase genome on average, that would be equivalent to a pitcher full of water compared to an Olympic-sized pool. So the equivalent of about 1 million bacterial genomes. That's a lot of data. And so that requires some unique ways to handle that data because you can't simply analyze it on your laptop computer as you would a bacterial genome. You need high-performance supercomputing. And so fortunately at Berkeley Lab, we've been able to collaborate and partner with the Microbiome project led by Kathy Yellick and her team shown here to use a tool called metagenomics, which is one of the first large-scale metagenome assemblers that can handle terabases worth of data and can run on the DOE's high-performance computing environments shown here, Perlmutter. And so what do you get when you get 5 terabases worth of metagenomic data, high-performance computing, and the right tools to handle the data? Well, you get a genome-resolved view of NEON's soil microbial communities, shown here. This is not a picture of Dippin' Dots. This is a picture of what the non-redundant 1,300 metagenome-assembled genomes look like from a bird's eye view. The color is the taxonomy, colored by taxonomy, and the bubble sizes are the samples that map to those reference mags. And so, as you would expect, we see many of the main soil microbial players across these samples. What we can now do, because NEON has such a wealth of standardized data, is that we can plot out and compare these soils across samples. So I'm showing here an ordination plot, which shows the different samples and how they compare to one another. And so not surprising, we see that within different samples, samples tend to cluster with one another. Now, what we can do next is overlay some of the important data that NEON collects to try to look at, say, for instance, carbon flux across these sites. So I have the same plot here shown overlaid with the average flux for carbon measurements that have been collected from these soil samples. And what is really exciting is that we can now combine this to a new view. So we have really robust environmental data from NEON. We have this metagenomic view from the NEON samples, and we can simply ask the question, how does carbon flux look from observed compared to the predicted? And this is important not just because we're interested in predicting, but presumably the prediction can lead us to information about the main drivers for how carbon flux is working within soil systems. So shown here in the top plot is when we look at observed versus predicted measurements using environmental parameters only. And in the bottom panel is when we incorporate this genomic information. We get an R-squared value of 0.95. I think that's pretty impressive, that using the genomic data can improve prediction of carbon flux within soil environments. So we can take this a step further. We can look at the same plot and connect the environmental information as such. So when we take, for instance, this top plot here with the highest CO2 value, we can zoom in on a particular site. We can investigate what mags were recovered from this data set. And then we can look in depth at the genome of particular populations. And so shown here is one of the more abundant mags within the Kanza site, which is actually a very rare soil bacterium, Roku bacterialis, which is uncultivated. Next, we can dive into that genome information and pull out important genes, such as shown here, carbon monoxide dehydrogenase. And this is important because we've now tied back from a genome to a gene and pathway level information, carbon monoxide being translated to carbon dioxide. And so we've taken this now full circle. So this is, again, just a pilot and a story demonstrating that with improved methods, we can better understand how environmental microbiomes can feed into carbon flux and tracing carbon flux across terrestrial systems. So this is an example of what can be accomplished when large-scale facilities collecting standardized and using standardized methods can come together. But this is also a very US-centric example. So, of course, we have terrestrial systems around the world and scientists around the world. So can we do the same thing with existing data that has been made publicly available? So NCBI is the primary data repository for sequence data. And what we're interested in looking at is the available data that could be used and reused. When we look specifically at the biosample records, there are 37 million records that were recovered back in February of this year. A much smaller subset of that data, if we're interested in looking at soil metagenome samples, was data that was submitted using this generic record. And then a very much smaller amount of data, specifically 115,000 records, is soil metagenome biosample that was categorized and submitted using the Genomic Standards Consortium template for that type of data. I will say that there is likely a lot of soil metagenome data in this generic record, but we wouldn't know that because it would take human curation to dig into that data and try to pull out that soil metagenome information. So we're looking at about less than 1% of data that we would be able to use just off the get-go. When we want to say, look at this data even further, and we're looking at this 115,000 soil metagenome biosample categorized records, we might want to just take that data and use it. Do you think we can do that? Yes? No? All right. The answer is no. We are funneling down into an even larger or smaller subset of data, 78,000 records that are machine actionable. What does machine actionable mean? That means that data can be used to input into new machine learning approaches, but otherwise data needs to be curated in order to use it. And that misformatted data is really a problem. So if we zoom in at an example, I'm showing here one of the required categories for soil metagenome data, depth. So it's important to know at which depth do you collect a soil sample. When we look at this, the Genomic Standards Consortium requirements are that you have a number and a unit. And here are a couple examples of information that's been misformatted. So the top bullet is presumably someone submitted latitude, longitude coordinates, but in the wrong category depth. Or there's a whole range of series of measurements with or without units, or the second one, my favorite, centimeters, presumably centimeters. This is a problem, and I'll say for my dear NCBI colleagues out there, this is not a commentary on NCBI. This is a commentary on us as a research community that we need to do better. We really, thank you, Jonathan. We really must do better. And so the value of all those 37 million biosample records is really limited. So where do we go from here? Well, I come from the perspective that researchers actually want their data to be useful and used and reused beyond the primary reason why that data was generated. And so let's start by making it easier for researchers to submit their data and to make it useful for the rest of us. And so our team at the National Microbiome Data Collaborative has been working on just that. We've been working on a mobile app, since all of us like to be on our phones and use our phones all the time, to improve how we can collect data in the field. And so here I'm introducing the NMDC Field Notes app. This is a tool in collaboration, in technical collaboration with the Genomic Standards Consortium, where we've enabled use of those standards in a streamlined and standardized way within this format. So whether you're an oceanographer, whether you're a soil scientist, you can use standardized templates in the field and streamlined so you don't have to deal with later organizing your data and perhaps not being able to standardize. We use basic tools for easy data entry that is compliant and conformant with the standards themselves. We can use GPS coordinates, so we don't have to deal with formatting the latitude and longitude. One of the nice aspects of this mobile app is that it's consistent with our submission portal. So whether you have internet connection or whether you don't, you can collect this information locally on your phone and submit it to the NMDC submission portal to be completely standardized. We think that this app is going to really lower the barriers for being compliant and providing all of the metadata information that is later hard to do once you've come from the field and once you are writing up your publication. So we're really excited about this new tool. We will be providing early access to the Field Notes app in fall of this year. Right now, we're looking for volunteers, all of you, to be beta testers, to test this out in the field, to give us feedback, and to iteratively work with our team to make sure that this is something that will be useful for your research. And so with that, I just want to thank my team, the incredible people that I get to work with. Our foundation, our collaboration is really based on collaboration, and I'm so fortunate to work with these amazing people. So thank you.
I'd like to start by saying thank you to the organizers for a wonderful conference. Thank you to Virginia for everything you've done over the past year. And thank you to all of you for joining me here. As I start the talk, I want to actually ask you to remember some unpleasant things about the past 4 years. So we all recognize the devastating impact of COVID-19. In just the past 4 years, there were 3 million lives lost. That was just before vaccines were available, along with 23 million jobs in the United States alone and $14 trillion in economic damage. Any of you who are parents there probably recognize the terrible impact of the pandemic on our children's education. We currently are dealing with an estimated 65 million cases of long COVID or lingering effects from COVID-19 infection. And a point that really hit me when I looked at the numbers around the past pandemic was the fact that there have been more deaths from COVID-19 in the United States than all combat deaths from the history of the United States. So as we look at this, we know this was a terrible event. But I want you to think about now is imagine how different this could have been if we were actually prepared. And we need to be prepared. I'm here to tell you now that the next viral pandemic is inevitable. This will happen again. In the 21st century alone, we've already seen multiple viral pandemics. And we know that new viruses are continuing to emerge with increasing frequency. We've all been incredibly grateful for the work done to generate vaccines. But at least with current technology, vaccines are reactive. Vaccines can only be developed when a pathogen starts to spread widely and the sequence is available. In the meantime, we need a defense. We need antiviral drugs as that first line of defense to reduce disease and prevent hospitalization and death while vaccines can be generated. So if we know the next pandemic's coming, what should we start preparing for? The truth of the matter is we don't know what virus comes next. However, there are a few things about viruses that we can leverage to start preparing. The first is that based on their genetic relatedness, viruses cluster into families. Now, viruses in a family all share common features that can actually be targeted by antiviral drugs. We also have a very good sense of which virus families have pandemic potential. So while we don't know which specific virus comes next, we do know which families are likely to emerge from. So we can leverage that information to start making the antiviral drugs we need right now, and in particular, broad spectrum antiviral drugs. I want to define a couple terms first. By broad spectrum, I mean drugs that are effective against multiple viruses in a single family. A drug that's effective against all viruses, we would call a panviral drug. The other thing is we're focused on small molecule drugs. So these are low molecular weight organic compounds. Think aspirin, not biologics. Now, the advantages of these broad spectrum antiviral drugs are actually twofold. First, the majority of diseases that cause, or viruses that cause disease in humans today have no approved therapies and no approved vaccines. So a drug that works against an entire family of viruses could actually provide the first treatment for viral diseases that sicken and kill millions of people around the world today. But if a drug works against multiple viruses in a family, it's highly likely to work against the next new virus that emerges in that family. So in addition, these broad spectrum drugs provide protection from future threats. When we're thinking about our therapeutics, we need to think about treating everyone around the world. So that's why we focus on orally bioavailable drugs, or pills, that we can actually ship immediately without refrigeration to anyone, anywhere in the world, to help reduce disease, prevent hospitalization and death, and slow the spread of the next virus until a vaccine can be made. So there's broad agreement that this is an absolutely critical approach to preparedness for the next pandemic. Small molecule antivirals are considered to be the most effective treatment for a new virus and really need to be the cornerstone of our pandemic preparedness efforts. Broad spectrum, again, provides us with that immediate preparedness, immediate response on the first day that a new virus emerges. And just to give you an example of the kind of impact a drug on day one could have, if only 1 out of every 5 symptomatic COVID-19 patients had taken a drug like Paxlovid during the 2022 SARS-CoV-2 Omicron wave, we could have prevented 33,000 deaths and 850,000 hospitalizations at a cost of over $170 billion. So if we have broad consensus, why isn't anyone doing this? There's really 3 major gaps to solving this problem. The first is what we call the discovery gap. So there are lots of folks out there in academic labs around the world that are working on solving this problem of finding new antiviral compounds, that discovery and initial development phase. However, academic labs don't typically have access to the resources that you need to actually move a promising inhibitor into preclinical and later clinical development. Now, on the other hand, pharma companies do have those resources and that technology at hand. However, businesses develop drugs to target specific indications for existing viruses to a market. That doesn't make pharma companies bad for not being involved in this research. It just makes them businesses. However, of course, there is no business model to fight diseases that don't exist yet, like the next pandemic virus. And really, that is the motivation that led myself and my 2 co-founders, Drs. Ralph Barrick and Mark Heisey, to found READDI, which is a mission-driven, nonprofit biotechnology company that's focused on making broad-spectrum, small-molecule antiviral drugs for viruses that cause pandemics. A key piece of this mission is our commitment to global access, making sure that everyone, everywhere, has access to the drugs that we seek to make and can afford them. And along the way, we're helping to address the market failure that many of you who work on things like AMR understand that limit development so that we can disrupt this panic neglect cycle of pandemic response. So we're focused on 8 virus families that are known to have pandemic potential and are the likely origin of future disease X or new virus. Some of these virus families, I'm sure, are familiar to many of you. Coronaviruses need no introduction at this point in time. But we're also focused on families like filoviruses, which include Ebola virus, flaviviruses like Zika virus and dengue virus, which is currently causing a major epidemic in South and Central America with millions of cases, as well as viruses like the orthomyxoviruses. And I I'm sure that many of you have heard of the looming threat of H5N1. Right now, we're working on 5 of these families with plans to expand to the remainder. And I put this slide together to really drive home the urgent need we have to develop these drugs. So on the left is a graph of the number of compounds or assets that are currently in clinical development for 6 of these virus families. And on the right is a graph of any therapeutic modality for any of these viruses at any stage of development. And the piece that's striking and that's saddening to me is the coverage bare. So I want us to set aside coronaviruses for a second and look at these other 5 virus families. Some of these virus families have almost nothing in clinical development. And those of you who have been involved in drug development in the past recognize that the number of clinical candidates that are being explored now is likely not enough to actually end up with an improved therapeutic. The situation's not much better when we go further back in the development pipeline and look at discovery efforts and preclinical efforts. There's really just not much work being done. But there is good news. If we go back to the coronaviruses, what we can see is that success is possible. In 2019, that coronavirus graph looked just like the toga virus and the arenavirus numbers. But with effort, expertise, and resources, we can solve this problem. And we all know this in the room because we have antiviral drugs for coronaviruses today. So that's where READDI steps in to bridge this gap. There's really 2 main efforts that we're involved in. The first is an accelerating discovery. So bringing together teams of researchers from around the world to do that critical foundational research to discover those antiviral compounds and start to move them forward. We take a high-risk, rapid failure approach. When you're looking for new small molecules, most of them won't work out. So we try to kill them as fast as we can so that we can move on and focus our energies on the most promising compounds. But the second area of activity is really driving these new compounds towards further development. So collaborating with the drug innovators themselves to help guide the advancement of these compounds towards the clinic, and then going out and raising the funds to help de-risk those assets and move them towards clinical testing. I'm just going to give you one quick example of the kind of the work that we're doing right now. This is the program I'll talk about is focused on toga viruses or alpha viruses. So these are epidemic and weaponized viruses that cause widespread disease. 2 examples that you may have heard of. The first is called chikungunya virus, a mosquito-borne pathogen that causes explosive outbreaks of disease. To give you an example, in 2013, there were no locally transmitted cases of chikungunya virus in the entire Western Hemisphere. By 2014, there were several million cases resulting from a single introduction event in the Caribbean. The other example in the toga virus family is a virus called Venezuelan equine encephalitis virus. This is a virus that was weaponized by both the United States and the Soviet Union during the Cold War. These viruses typically will cause an influenza-like illness, but viruses like chikungunya can cause a severe and sometimes chronic arthritis that can last from months to even years. In contrast, viruses like Venezuelan equine encephalitis virus, as its name suggests, can cause severe neuroincapacitation and encephalitis. Importantly, though, are the points at the bottom. There are no approved biologic or small molecule therapeutics for any toga virus. There is one limited use vaccine that was just approved a couple of months ago for chikungunya virus, not for any other toga virus. And of course, as we look forward to the next virus in this family that could emerge, again, no treatments are on hand. So how do we approach this problem? It's a very straightforward approach, very familiar to any of you who've worked in any kind of small molecule inhibitor development in the past. We picked a target. In this case, it's a viral protein, the NSP2 protease, which is absolutely essential for virus replication and disease. We chose this target for the toga viruses because we know that viral proteases have been successfully targeted for other disease indications like hepatitis C, HIV, and of course, drugs like Paxlovid for SARS-CoV-2. So we asked the simple question, can we develop an inhibitor for the NSP2 protease? So the first thing we did is we tracked down a bunch of experts in viral proteases, biochemists, to help us establish an assay. In this case, we established a FRET-based assay to measure protease activity. We then used that assay to screen over 117,000 compounds from a variety of different inhibitor sets to find some promising starting points, one of which turned out to be a quite promising starting point, a compound that can inhibit the protease with an IC50 of around 39 nanomolar. But perhaps more importantly, this compound can also inhibit virus replication, not just of chikungunya virus, the original target, but also of Venezuelan equine encephalitis virus. We've just recently moved this forward to show that it could in fact inhibit virus replication in vivo. So now we have proof of concept that our approach could be useful in developing this compound further to becoming an antiviral drug. And in fact, we're in that full-blown lead optimization and preclinical development program now. And I'll spare you the details of our safety, toxicology studies, and manufacturing process. So I'm a virologist. I'm a basic scientist by training. I don't know how to make a biochemical assay. I don't know how to run a high-throughput screen. But what I did do is go out and find people who are biochemists and who do know how to run screens and test enzyme activity, and who are medicinal chemists who can optimize that compound. I play a very small role in any of this. As you can see, it took a team of individuals at 7 different universities to get us to where we are. Now think back to all those different virus families and all the different targets that we need to address. And that's why we've assembled a large coalition of both academic, biotech, and pharma partners that are working together in teams on all the different targets for all these different virus families. Based on this work and the coalition that we've built, we're very excited to serve as a core implementation partner for something called the 100 Days Mission, led by the International Pandemic Preparedness Secretariat. If you haven't followed the work of the IPPS, I encourage you to check it out online. This is a group that it was launched by the G7, led out of the Wellcome Trust, that's really focused on the 3 pieces that we need for pandemic preparedness: diagnostic development, vaccine development, and of course, therapeutic development as well. So this is our pipeline right now. One of those bars in the toga virus is the example I showed you. But as you can see, these are the 5 families that we're working on and the different programs that team of people I showed you is working to push forward. But the other thing I want to tell you is this isn't good enough, not even close. If we actually want to be prepared for that next virus that I know is coming, and I hope you do too, all of these bars need to be to the far right side. We need to have drugs, at least 2, for each of these virus families, what we call phase 2 ready. So through phase 1 human safety testing with efficacy established in preclinical models, so that the very first day we know that virus is circulating, we can launch our efficacy testing. We'll know how to manufacture that drug. We'll know how to distribute it, and we'll know how to dose it so that we can stop that next catastrophe. So this was all the faces I could fit on this slide. There are more, but these are all the lab heads from the different organizations that are involved. I actually talked to our program manager about putting together a slide that had all the names of the individual researchers. She encouraged me not to do that because it's simply too many. It's actually a little bit over 170 different individuals that are taking part in this effort. Of course, I also would like to say thank you to all of our funders. They've been very generous, and to date, we've raised a little over $90 million to help support this effort. Again, to those of you involved in therapeutic development, you know that $90 million is a drop in the bucket for what we need. And in fact, just to move that pipeline I showed you before all the way to phase 2 ready will require at least $500 million additional dollars. So with that, I want to end with a request. I want to ask any of you who have the skills, the expertise, and the knowledge that we need to help us get ready for the next pandemic. We need biochemists. We need medicinal chemists. We need experts in formulation and pharmacology, toxicology, and of course, we need a few virologists as well. But for any of you who would be interested in helping in any way, I would encourage you to visit our website where there's a contact form and you can reach out. Our project teams are always forming and always need additional help. And also, I'd invite you to follow us on LinkedIn to keep track of our progress. With that, I'd like to say thank you again to all of you for listening, and I will hand the podium back to Virginia.
And now it's my great pleasure to sort of hand over the baton to our incoming president of ASM, Terry Kohler, who's going to tell you about her vision for her President's Forum. Welcome, Terry.
So one of the really wonderful opportunities that I'm going to have as your president is I get to plan next year's Microbe President's Forum. And so I want to give you a little bit of a teaser tonight on what that's going to be about. Next year, we're going to be talking about AI and machine learning at our forum. And I think you'll agree with me that this is a very timely topic for us. I don't know about you, but in many of the talks I've heard so far at this conference, there's been a lot of examples of use of and discussion of machine learning. We even heard a little bit about it tonight in a couple of our talks. So I have to also tell you that one of my gifts is not having the ability to make great titles for forums or seminars or what have you. And so in thinking about this forum, I knew I have a vision, I know what I want to do, but I couldn't come up with a great title, right? So I decided that I would ask ChatGPT for some titles for this. I thought that was appropriate. And so I fed in my question with some key words, and the first thing I got was exploring the intersection of machine learning and scientific research in microbiology. I thought that was pretty good, but I wanted to have some more options. And so I asked again, and I got innovations at the nexus. Well, that's kind of a cool term. Machine learning, scientific research, and education in microbiology. And then I was kind of having some fun. So I said, well, give me a more dramatic title that'll really attract people. And I got microbial marvels, right? Integrating machine learning for cutting edge research and training. And then the last thing I did before I wasted my entire afternoon on this was I said, well, maybe give me a title that's kind of witty, right? Let's have a little humor here. And I got microbial minds and machine learning, cultivating intelligence in petri dishes and pixels. So we're not going to go that far. So I decided on the first title that ChatGPT offered, which is exploring the intersection of machine learning and scientific research in microbiology. And I'm very excited about planning this forum. And I hope to see you all next year at the forum in LA. And so with that, have a great rest of your evening, and thank you for coming.